Hello everyone. So from now onwards, I'm gonna upload around 11 parts for the crash course of the physiology, and in this 11 parts, I'm gonna complete the whole syllabus for the physiology. So over here in the part one, the first chapter, that is the general physiology. So the most common question over here, the cell structure and the function. So you all know about the cell structure and the function, the organelles inside the human cell. So let's have a quick recap of this. The nucleus, nucleolus, ribosomes, endoplasmic reticulum, that is the smooth and the rough one. The rough ER is also known as the granular, and the smooth also known as the agranular. Golgi apparatus, mitochondria, lysosomes, secretory vesicles, microtubules and the filaments, and the centrioles. Just have this quick recap of this, the structure, the function of this. I guess you all know about this, right? Okay, so one important thing over here is that the Tay-Sachs disease. Now, what is this Tay-Sachs disease? It is the congenital absence of any one of the lysosomal enzymes. So if you remember, the lysosomes are present over here in the cells, okay, and their main function is to break down the worn out cellular components or engulfed particles. So over here there is, in this disease, there is congenital absence of any one of the lysosomal enzymes. So what will happen? There will be accumulation of the substrates in the lysosomes which they normally degrade, okay, and these finally disrupts the cell function that contains the defective lysosomes, leading to the lysosomal storage disease known as the Tay-Sachs disease. Now next comes the types of the proteins present in the cell membrane. So firstly we have the structural proteins, including the integral and the peripheral membrane proteins. The next we have is the transport proteins. Now these transport proteins includes the channels and the carriers. So in the channels we have further continuously open type of channel proteins or channel transport proteins that includes the water channels. Then gated channels. In the gated channel we have further more types of channels, including the voltage gated channels, for example the sodium channels. Ligand gated channels, and these ligand gated channels are further divided into two types, that is the external and the internal. Now next we have is the mechanical channels, and they are present in the hair cells of the organ of Corti. The next are the carriers, carrier transport proteins. So they include the uniport and the symport and antiport. Okay, uniport includes the example, that is a glucose transporter. Symport includes the absorption of the sodium and the glucose in the intestines and the kidney tubules. And the antiports include the sodium and the calcium exchange in the cardiac muscle. So over here we all know what is uniport. That means over here only one type of substance is passing through the channel in the one direction. What is symport? Over here in the same directions, two different substances are going through the channels, and in the antiport, two different substances are going through a single channel in the opposite direction. The third, pumps. Pumps are the enzymes with ATPase activity which actively transport the ions across the cell membrane, include the sodium-potassium pump. The next is the receptors. They are bind to the specific ligands like hormones and the neurotransmitters. Enzymes, they are catalyze the chemical reactions. Glycoproteins for the antibody processing and the self-recognition from the non-self. And the cell adhesion molecules, they anchor the cells to each other and the basal lamina. So these were the types of protein present in the cell membrane. So these can be asked in around 2 to 3 marker. Now next here is the intercellular junctions. One more important question. Now in the intercellular junctions, we have first of all tight junctions. The tight junctions, they are formed by the ridges between the cells, which are formed by the fusion of the cell membrane of the adjacent cells. Okay, and they forms a belt between them. Okay, so over here we have these as the tight junctions. Tight junctions over here. Now such junctions prevent the transport of the substances between the cells. And such type of junctions are located in the epithelium lining the intestinal mucosa, walls of the renal tubules, and also the choroid plexus. These junctions prevent the passage of the substances from the lumen to the epithelial cells. Now next we have is the adherens junction. They mediate the cell-to-cell adhesion via the action of the nectins and the cadherins. The next is the desmonems. Desmonems are the button-like junctions, and they are present between the cells through the thickened spots on both the cells, and their membranes do not come in contact at the desmonems. At the site of the desmonems, towards the cytoplasmic sites are the filamentous structures radiating from the thickened membrane. So here you can see these are the cytoskeletal filament. Desmonyms hold the cells together and also attach them to the basement membrane, providing the strength and the stability to the tissues. Now finally we have the gap junctions. At the gap junctions, a 2 to 3 nanometer gap exists between the adjacent cells. So over here you can see in this diagram the cell, there is gap junction between the cell, and over here the gap is traversed by the hollow cylindrical channels formed by the regular arrangements of the protein units, that is the connexons. Now next it is the membrane potentials and the resting membrane potentials of a cell. So over here the magnitude of the resting membrane potential varies between the -10 and -100 millivolt. So over here first of all you should know that in many body cells, especially the excitable tissues, that is the nerve and the muscles, a potential difference is present across the cell membrane when the tissue is at rest. Okay, so at rest also there is some sort of potential difference in the across the cell membranes. Okay, and this is known as the resting membrane potential, as the name says. So inside of the cell, the RMP is negative in relation to the outside. So that's why you should remember that inside the cell, the RMP is negative as compared to the outside. So that's why the magnitude of the RMP varies between -10 and -100 millivolts. Now over here, the minus sign signifies that it is negative inside, as I told you before also. In the nerve, the RMP is around -70, and in the skeletal muscle, it is around -90 millivolts. You should remember this units, okay? How the RMP is generated in the cell. So there are two main reasons. That is the selective permeability of the cell membrane. Okay, so it is permeable to potassium and impermeable to protein and ions. And also activity of the sodium potassium ATPase pump present in the cell membrane. So what it will do? It pumps out 3 sodium and 2 potassium enters the cell. So 3 sodium goes out and 2 potassium comes in. Now next it is the equilibrium potential. So the potential difference across the membrane at which there is no net movement of an ion, that is influx and the efflux are equal. That is the equilibrium potential, as the name says. Equilibrium means equal. Now what is guiding these ions movement? So movement of the ions across the cell membrane is guided by the very important 2 factors. That is the concentration gradient and the electrical gradient. Now what is concentration gradient? It is occurring due to the difference in the intracellular and the extracellular concentration. This concentration gradient occurs, which causes the movement of the ions across the cell membrane. And next it is the electrical gradient. Inside of the membrane is negatively charged and favors entry of the anions towards inside and opposite that of the cation. Simple. Now there are 3 important ions of the physiological importance. Now what are these 3 ions? Just answer me fast. So these are 3, that is the sodium, potassium and the chlorine. Yes. So first of all, the potassium. The concentration gradient favors the efflux, whereas the electrical gradient favors the influx. Sodium, concentration gradient directed towards the inside and the electrical gradient is also in the same direction. And the chlorine, concentration gradient favors the influx, whereas the electrical gradient favors the efflux. Now next, very important, there is the Nernst equation. The magnitude of the equilibrium potential can be calculated from this Nernst equation. That's why this Nernst equation is very important. Okay, this is the Nernst equation. Just remember this whole formula over here. This is the Nernst equation. Next, it is the factors that generate the resting membrane potential. The difference in the concentration of the potassium in the extra and the intracellular fluid is very important for generating the membrane potential, the resting membrane potential. The next, it is the permeability of the membrane to the protein and ions. Membrane is poorly permeable to the sodium. We discussed about this before also, right? Activity of the sodium potassium ATPase pump in the membrane. And next, it is the magnitude of the RMP can be also calculated by using the Goldman constant field equation. Okay, so this is the whole equation over here. Over here, the P denotes the permeability for the potassium, sodium and the chlorine ions. The next very important question, how the structures are transported across the cell membrane. So there are many processes. So first of all, we have the very simplest one, that is the diffusion process. It is a physical process and occurs along the concentration and the electrical gradient. Solvent drag. Bulk flow of the solvent drags the solute molecules along with it. Then the osmosis, again important, movement of the water across a semi-permeable membrane. Okay, so you should know the difference between the diffusion and the osmosis. Now next is the most important one, there is a carrier mediated. Now why it is important? Because with the help of this carrier molecule, the speed of the transportation is increased by many folds. And it is of further two types, that is the facilitated and the active. Now the facilitated is a type, or we can say it is a form of passive transportation that does not need energy as it occurs along with the gradient. Okay, and it is carrier mediated process that enables the molecules that are too large to flow through the membrane channels by simple diffusion to cross the membrane, and it is much faster than even simple diffusion. Okay, and the transportation is along their electrochemical gradient, that's why it is faster. The next we have it is the active transportation, and as the name says, active transportation, so it will be requiring the energy and occurs against the concentration gradient, that's why it requires energy in the form of ATP. Now next, it is of further two types, there is the primary active transportation and the secondary active transportation. Now filtration, fluid under the pressure is forced through the membrane, for example glomerular filtration is very common. Endocytosis and the exocytosis, for example secretory granules. Now active transportation ask as separate questions, just pay attention over here. Active transportation refers to the mechanism of the transportation of the substances against the chemical and or electrical gradient. So that's why as it is going against the gradient, so it will be requiring ATP as the energy form. It involves the expenditure of the energy which is liberated by breakdown of the higher energy compounds like ATP. The active transportation is of further two types, that is the primary and the secondary one. Ask as separate questions, very important. Okay, so primary active transportation. The energy is derived directly from the breakdown of the ATP or some other high energy phosphate compound. So where the main energy form it is the ATP. Examples are very important, the sodium potassium pump, very important, and the calcium pump. So over here, very simple video is over here, which is showing the sodium potassium ATPase pump, how they actually work. Okay, now next it is a very important, the secondary active transportation. Over here the energy is derived secondarily from the energy that was stored in the form of ion concentration difference between the two sides of the membrane created in the first place by the primary active transportation. There is no difference between these two. Okay. Now at many areas in the body, the transportation of some other substances is coupled with the active transportation of the sodium, and they are known as the sodium co-transporter or counter-transporter. These may occur in the form of two, sodium co-transport and the sodium counter-transport. Now sodium co-transportation of the glucose. Co-transport, that means they are going together. Co-transport across the cell membrane is a type of coupled transport mechanism in which two substances are simultaneously transported across the membrane, and it requires a carrier to which both the substances must be bound for the transportation. As the name says, co-transport means going together. It requires energy in the form of ATP. In this system, one of the substances is transferred with the gradient and the other against the concentration gradient, but they are having the co-transportation. Okay. Example includes the mechanism of the absorption of the glucose or the amino acid across the intestines along with the sodium. Now as over here you can see, they are having the sodium and the glucose. They are going in the same direction, the co-transportation occurring. Sodium, glucose, co-transportation. Now next we have the sodium counter-transport. Okay, counter means in the opposite directions. As one is coming inside, the other is going outside. Antipode carriers are used, that is the sodium ion is exchanged for some other substances. So that's why they're also known as the exchangers. Okay, they're exchanging a particular substances, okay, with the sodium. Okay. Example include the sodium calcium counter transport, and that occurs in almost all cell membranes with the sodium ions moving in and calcium ion going out the cell, as you can see in this diagram also. The other example include the sodium hydrogen exchanger, and it occurs in the proximal tubules of the kidney. Now let's compare the primary and the secondary active transportation, because it is mostly asked in the exams, okay, as a very 3 or 4 marker questions. Okay, so let's have this. Primary, over here the transportation of the molecules occurs against the concentration gradient by the use of energy from the ATP, okay, directly from the ATP. And the secondary, the energy is derived secondarily from the energy which has been stored in the form of ion concentration difference between the two sides of the membrane created in the first place by primary active transportation. Just remember these two definitions, they are very important. The primary, the transmembrane proteins are unique to the ion transported through them. So the transmembrane proteins are unique over here. And also the examples include the sodium potassium pump, calcium pump in the muscles, proton pump in the stomach. Now next we have the secondary active transportation. And over here the symphodes and the antipodes are different types of transmembrane proteins. Okay, there is no specific type of transmembrane protein over here as there was in the primary one. Okay, in the secondary one we have symphodes and antipodes as we have discussed earlier. The examples are very important. There is a glucose sodium pump. I showed you the video of this also before, right? Sodium calcium exchanger, sodium hydrogen counter transportation. Okay, so very important, diffusion. Diffusion is passive transportation, a process by which molecules move from an area of high concentration to area of low concentration. Simple. And in other words, we can also say that cations move to negatively charged area, whereas the anions move to positive charged area. Now types of diffusion, very simple. Simple diffusion and the facilitated diffusion. And over here in the facilitated, we discussed previously also that it includes the carrier proteins. Example, the GLUT. Now what are the 3 main differences between these two types of diffusion? Now these 3 main differences include the specificity, saturation and the competition. Specificity, the carrier proteins. Okay. Saturation. In the simple diffusion, the rate of diffusion increases proportionally with the increment in the concentration of the substance, and there is no limit to it. However, in the other diffusion, the rate of diffusion increases with increase in the concentration gradient. That's why saturation over here is also a main difference. The next, it is the competition. When two molecules are carried by the same protein, there occurs a competition between the two molecules for transportation. And remember over here, such competition do not occur in the simple diffusion. That's why these 3 are the main difference between these two types of diffusion. Okay. Now next, what is homeostasis? The tendency of an organism or the cell to regulate the chemical processes that takes place internally so as to maintain the health and the functioning, and these all are regardless to the outside condition. Okay, so we have over here, now to maintain the homeostasis, we have various feedback mechanism in the body, including the positive feedback mechanism and the negative feedback mechanism. So the positive one, it results into amplification or the growth of the output signal, as the name says. Also, it strengthens the stimulus and increases the productivity, and it is less associated with the stability of the system. While the negative feedback homeostasis, it results into inhibition, as the name says, or slowing down of the process, and it reduces the stimulus effect and decreases the productivity, and it is more associated with the stability of the system. Now, the most important question, what are the examples of the positive and the negative feedback homeostasis? So here, for example, there are 2 main examples of the positive feedback mechanism. There is a childbirth and the blood clotting mechanism. And for the negative feedback, the body temperature and the blood pressure are a very important example. As the body temperature increases, by the negative feedback, it comes into the normal human temperature, the human body temperature. And for blood pressure also, whenever the blood pressure increases, by the negative feedback, it is come back to normal. Now next, it is the hormonal regulation of the blood volume. Now you might be thinking that this is not an important topic, but it is, because it comes under the homeostasis topic only. Because what was in the homeostasis? There is a maintaining of the internal environment inside the human body, regardless of the external environment, right? So over here, blood volume plays a very important role. The water inside the body, the fluid inside the body plays a very important role in maintaining the internal environment inside the human body. That's why hormonal regulation of blood volume comes under topic homeostasis only. So over here, feedback mechanism occurs for the controlling of ECF, extracellular fluid, blood volume, cardiac output, and the arterial blood pressure. So over here, for the controlling of the ECF, it involves the control of the two factors, that is the ECF volume and its tonicity. For controlling of the ECF volume in the body, the sodium balance should be done in the body. And for balancing the sodium in the body, this sodium balance is controlled by, first of all, the kidney. The sodium ion is filtered in the kidney at the glomerulus and a constant fraction of it is reabsorbed in the proximal tubules known as the glomerulotubular balance. Aldosterone influences the reabsorption of the sodium at the distal convoluted tubules. You all know. Aldosterone secretion is controlled by the renin-angiotensin system, whose secretion is in turn controlled by the level of the potassium and the sodium of the ECF. Now, next important mechanism, it is the ANP, that is the atrial natriuretic peptide. Now, it is secreted from the atrial muscles and promotes the excretion of the sodium through the kidney when the ECF volume is increased in the body. Now, here this mechanism shows a very clear role of the ADH, anti-diuretic hormone. Whenever the ECF volume is increased in the body, whenever this ECF is increased, what will happen? The blood volume increase and this atrial stretching occurs. It is also increased. That's why a reflex inhibition of the ADH release occurring. Okay, so here there will be inhibition of the ADH release, and this will in turn cause less water reabsorption in the kidney and more water loss, and this will lead to decrement in the ECF. So we can consider this in the negative feedback mechanism also. Over here, what is happening? There is inhibition of the ECF volume, decrement in the ECF volume by the way of inhibition of the ADH release. Now the control of the tonicity of the ECF, this was the controlling of the ECF volume. Now control of the tonicity of the ECF. So over here, the regulatory mechanism includes the hypothalamic osmoreceptors and thirst mechanism. Again, a very important question commonly asked in the exams. So over here, first of all, the osmoreceptors, they act by controlling the ADH secretion, and these are neurons in the hypothalamus, and they are sensitive to changes in the osmolarity or the tonicity of the interstitial fluid around them. Now next, it is the thirst mechanism. The thirst center is located in the lateral hypothalamus, which when stimulated, there is conscious desire to drink water, and the neurons are stimulated when the osmolarity of the interstitium increases. Then next, it is the Starling forces. Again, a very important concept, not only for exams but also for your practical purposes also. Okay, and these forces are just understand over here, and this will be important in almost every year of a study. So what is Starling forces? Capillary osmotic pressure across the capillary wall helps to maintain the fluid exchange at the tissue level. The rate of filtration and the absorption at any point along the capillary depends upon a balance of forces, which are called as the Starling forces. And over here, the four pressures which are causing the Starling force are involved. That is the, first of all, the hydrostatic pressure across the capillary wall contributing for the Starling forces. So over here, it is the hydrostatic pressure. Next we have it is the capillary osmotic pressure across the capillary wall. It is the plasma colloid osmotic pressure. Then next we have it is the hydrostatic pressure in the interstitial fluid. Okay, and next we have it is the interstitial fluid osmotic pressure. So these are the four pressures contributing for the Starling force. But remember over here, an important thing, that this factor 3 and factor 4. So these factors 3 and 4 tend to cancel out each other. Okay, and hence the forces which are actually determining the fluid exchange at the tissue level depends only on the first and second one. This first and the second one, because these two cancels each other. So factor 1 and 2 mainly contributes for the Starling forces. Okay. So normal ranges of the colloidal, this COP is the colloidal osmotic pressure, and the HP is the hydrostatic pressure. So commonly this COP is around 25 and this HP is around 15, that is 10 mmHg, and this will cause the absorption. Okay, but if this HP is increased from 15 to 37 around, means it is increased, and the COP is the same. Okay, so if this HP, the hydrostatic pressure, is increased, this will cause filtration process. Now from this important concept, there is very important applied aspect over here, that is the hypoproteinemia leading to edema. Now many times in exam it is asked that why the hypoproteinemia, that means decrement in the plasma protein, leads to edema. Okay, so what is the answer over here? So this answer is related over here through these Starling forces only, through these all HP and the COP. Now just understand, in the hypoproteinemia what is occurring? There is decrement in the plasma protein. Okay, and because of the decrement in the plasma protein, what will happen? This colloidal osmotic pressure will decrease. And I told you before also that this COP is decrease and this HP is increase. What will happen? There will be increment in the filtration process in the arteriolar end and decrement in the absorption in the venous end. And this increment in the filtration and decrement in the absorption at the venous end, what will happen? This results in the abnormal collection of the fluid in the interstitial fluid, leading to edema. That means increment in the filtration in the arteriolar end and decrement in the absorption in the venous end. This will lead to edema in the hypoproteinemia. So yeah, that's all for today. I hope I have covered all the important topics from the general physiology part, and in the part 2 I will be covering the blood chapter. And I hope you found this video helpful and this series also, this crash course series. So thank you so much.